Moving towards the topic conjugation. So in a simple form, I'll say that the conjugation is the process whereby a male or a donor bacterium, we call it the donor bacterium, or we can also refer the donor bacterium to male bacterium, that mates or make physical contact with a female or recipient bacteria. The recipient bacteria means that receive the genetic material from the male. We also call it, sometimes call it in simpler form, female bacterium, and transfer the genetic element to it. So again, the conjugation is the process by a male or donor bacterium mates or make physical contacts with a female or recipient bacteria and transfer genetic elements to it. So there are two processes by which the conjugation occur. There is conjugation between F plus and F minus cells, and there is the conjugation between HFR and F minus cells. So the before actually describing the conjugation between F plus and F minus cell and HFR and HF minus cell, I want to go towards the terminologies of those individual terms. So F plus means the F plus cells are those type of cell, those type of bacteria that contains the plasmid. In the bacteria without plasmid, the bacterial cells without the plasmid are known as F minus cells. So you have to remember the F plus cells are those cells that contain plasmids, and F minus cells are those type of cell which doesn't contain plasmids. And HFR, the HFR is termed as high frequency recombination. So HFR cell means high frequency recombination cells. High frequency recombination cells means the plasmid that is present in the bacteria is incorporated into its own genetic material. So these type of cells are HFR cells. So after knowing these terminologies, I want to proceed towards the different processes of the conjugation. So first of all, there is conjugation between F plus and F minus cell. What happened? There is one F plus cell and another is F minus cell. So between F plus and F minus cell, there is the contact between them, and that type of contact is incorporated by the 6 pilus. So 6 pilus is the bridge between the F plus and the F minus cell. So in the next step, what happens? In the first step, there is the physical contact between F plus and F minus cell. In the second step, what happens is that the F plus cells have the plasmids. You already know that, and those plasmid gets incorporated into F minus cells through the 6 pilus. You can see these plasmids move into the F minus cells, and this plasmid, after the incorporation of F plus cells plasmid into the S minus cell, the F minus cells becomes S plus cells because this plasmid is acquired by the F plus cells. So at the end of the F plus and F minus conjugation, the F minus cells becomes F plus cells. So again at the end, the F minus cells have plasmid, so it become F plus cells. Again I am going to revise you. So the F plus plasmid contains genes required for the 6 pilus and the conjugation. So the bacteria without this plasmid are termed at the F minus. So the 6 pilus on the F plus bacterium contacts the F minus bacterium. So the single strand of the plasmid DNA is transferred across the conjugation bridge. The conjugation bridge is the 6 pilus. So you have to know that actually in the conjugation between the F plus and F minus cell, there is no actual transfer of the chromosomal DNA. The chromosomal DNA remains intact in the both F plus and the F minus cell. There is no exchange of the chromosomal DNA. Only there is the exchange of the plasmids. Now let us discuss about the HFR and F minus conjugation. So what happens? There is the two types of cell, F plus cell and F minus cell. What happens? The plasmid of the F plus cells get incorporated into the genetic material. So that means we can say that the plasmid get incorporated into the chromosomal DNA of the F plus cells. So after the transfer of the chromosomal DNA, that means when the plasmid and the chromosomal DNA gets recombinant, That means the F plus plasmid become incorporated into the bacterial chromosomal DNA. The cell becomes HFR cell. The HFR cell means um, high frequency recombination cell. So we have the HFR cell and we have the F minus cell. So the HFR cell and the F minus cells conjugate each other. So there is the bridge between the F minus and the HFR cell, uh, and the genetic material of the HFR cell goes towards the F minus cells, and this genetic material gets incorporated into the F minus cell, so that becomes recombinant F minus cells. So at the end of the HFR and F minus conjugation, we have two cells. One is HFR cell and another is recombinant F minus cell. So this is the significance of the conjugation. We get entirely new two bacterium in the HFR and F minus conjugation. So again I am going to revise the HFR and F minus conjugation. The F plus plasmid can become incorporated into the bacterial chromosomal DNA. After the incorporation, it is called as a HFR cell. The HFR cell is high frequency recombinant cell. The transfer of the leading part of the plasmid and the few flanking chromosomal genes can occur through the physical contact. So the high frequency recombination may integrate some of those bacterial genes, and the recipient cell remains F plus but may have the new bacterial genes. So when they have the new bacterial genes, it is known as the recombinant F minus cell. Those recombinant genes are acquired from the HFR cells, and the HFR cell is formed when F plus cells plasmid gets incorporated into this chromosomal DNA. So this is all about the conjugation. Now let us move towards the process known as the transduction.